it's Ms. Fish, and I'm here to talk to you today about mutations. So, first off, we need to start with asking ourselves, what are mutations? Mutations are the permanent alteration of the nucleotide sequence of the genome of an organism. So what that basically means is that we change the DNA sequence of an organism. There are certain things that cause mutations. For example, DNA polymerase can make a mistake. It's very, very, very rare, but DNA polymerase may make a mistake and cause a mutation. Other things, such as chemicals like nicotine, can alter your DNA and make changes also. Radiation, such as X-rays and UV or ultraviolet radiation, which comes from the sun, can also alter your DNA. Most of the time we find results of these mutations, they form into cancers. So like skin cancer or lung cancer, those are examples of like what we see in our bodies as a result of mutations. So what are the types of mutations? Well, there are three basic types. The first is base substitution, the second is deletions, and the third is insertions. If you need more time, go ahead and pause now so that you can go ahead and finish writing and restart when you're ready. So, base substitution is the most common type of mutation. It alters a single point in the DNA. So what's happening here is we have one base coming in and taking the place of another. So for example, we have two types. Transition, which is when a purine, adenine, and guanine is substituted with another purine. So that means A is placed to G, and G can be replaced with A. That would be an example of a transition. This can also happen with pyrimidines. So when a pyrimidine, which is thymine or cytosine, is substituted for another pyrimidine, then it's also known as a transition. So basically it's whenever one of the types replaces one of the same type. Okay? Transversion is when a purine replaces a pyrimidine. So A or G will replace T or C, and vice versa, which means thymine and cytosine, or T and C, can replace adenine and guanine, A or G. Go ahead and pause now if you need to finish writing and restart when you're ready. So there's a few results of point mutations or base substitutions. The first is a silent mutation. The substitution codes for the same amino acid. So when you're looking at your codon chart, you will notice that there are some codons that code for the same exact amino acid. If our DNA was to be mutated and when it transfers into mRNA and it codes for that same amino acid, it would be silent. We wouldn't really see it, but there is a mutation there. Missense is the substitution codes for a different amino acid. It no longer makes sense. So M, make, miss, okay, and sense. Okay, so it no longer makes sense. There's a different amino acid. Nonsense is there's a premature stop signal. The protein essentially stops early. If you need more time, go ahead and pause now and restart when you're ready. Now we're going to talk about deletions, which is the next type of mutation. So deletions are when one or more bases are deleted. It causes something called a frame shift. A frame shift is when the bases are shifted either to the right or left and they no longer will be in groups of 3. The result of a deletion is that the DNA code shifts to the left for every base that is deleted. There can be more than one deletion in a sequence, so please be aware of that. Go ahead and pause if you need to and then restart when you're ready. Insertions. Additional bases are added to the DNA sequence. This also causes a frame shift. However, instead of moving to the left, when you have an insertion, the frames are shifted to the right because you're inserting a letter and everything will move to the right. There can also be more than one insertion in a DNA sequence. Go ahead and pause if you need to now so you can finish writing. So, the big question, are mutations good or bad? Well, mutations can be good, but some can be bad too. It all depends on how the code for the protein was changed. For example, if you've ever heard of sickle cell anemia, that is when your blood cells are in the shape of a sickle, which is like a quarter moon shape. Okay? And what happens is when your blood is in that shape, your body can't carry the oxygen. So that's one way a mutation is bad. Another way a mutation could be good is if, let's say, we have a species of animal who lives in an environment and let's say their coat changes with their further generations. If that coat allows that animal to adapt and survive longer, that'd be a good mutation and they would be able to continue on living. If a protein ends, it is not fully formed, and that is not a good thing either. So it would be bad if your proteins do not get formed fully. Well, I hope you guys have a great day. Let me know if you have any questions, and I will see you in class. Bye!